We are going to continue. Next heading is chitin. It is there in your syllabus. Chitin is a linear homopolysaccharide. Chitin is a linear homopolysaccharide, and all the monomeric units are bonded together by means of beta 1,4 glycosidic bond. And then what's the difference between chitin and cellulose? It's very simple. Cellulose is made up of beta D glucose, while in chitin, of course, glucose is there, but it is modified. We call it N-acetyl glucosamine. The monomeric unit of chitin is N-acetyl glucosamine. Let us go to a picture. This one. This is basically glucose. Is it alpha D glucose or beta D glucose? Look at the OH group on carbon number 1. It is upward. So it is beta D glucose. And it was glucose amine because of this NH2 group on carbon number 2. It is glucose amine. But in that glucose amine, one of the hydrogen is replaced with an acetate group. So we say N-acetyl beta D glucosamine. This compound is N-acetyl beta D glucosamine. You see, all these units are N-acetyl beta D glucosamine, and they are joined together by means of beta 1,4 glycosidic bonds. And how what's the importance of this chitin? Chitin is present. Is it given here in this text? Yes, chitin is present in the cell walls of algae and fungi and yeast. Especially in fungus and all, chitin is present. And it is also present on the exoskeleton of some insects and crustaceans. I will show you a wonderful picture. Have you seen this insect? Especially in the coconut plantations and all, you might have seen such insects. It is having a very hard exoskeleton. An armored plate is there, This armored plate, this hard skeleton is made up of chitin. Okay? Yes. Now we move on to heteropolysaccharide. What's a heteropolysaccharide? It's a polysaccharide made up of more than one type of monomers. A good example is hyaluronate, hyaluronic acid. Actually, hyaluronic acid is present as a lubricant in the synovial fluid of joints. In your joints, in your body, you have synovial fluid. In that synovial fluid, actually that synovial fluid is lubricant. And a very important component of that synovial fluid is hyaluronate. Another one, in our eyes, in our eye, you have aqueous humor, vitreous humor, etc. In that vitreous humor, there is a very good amount of hyaluronate. And it is also present in the extracellular matrix of cartilage and tendons. Cartilage and tendons. And it is providing this cartilage, tendons, etc. with tensile strength and elasticity. Okay, so this is biologically, biochemically very much important. And what is it formed of? You see here, this is actually, what is it? It is glucuronic acid. Glucuronic acid. It is not alpha, but it is beta. So beta D glucuronic acid is one of the component. Look here. Beta D glucuronic acid is one of the component. One of the monomer. The other monomer is glucose. It was beta glucose. It was glucosamine. It was N-acetyl glucosamine. Look here, N-acetyl glucosamine. N-acetyl beta D glucosamine. So one of the component is beta D-glucuronic acid. The other component is N-acetyl beta D-glucosamine. And what is the linkage? Linkage is between carbon number 1 and carbon number 3, not with carbon number 4 of the N-acetyl glucosamine. So the linkage is beta 1,3 linkage. And this is hyaluronic acid. A polymer. Hyaluronic acid is actually a polymer of these monomeric units. And heparin. In your lower classes itself, you might have heard of heparin. You know, heparin is an anticoagulant. In our body, heparin may be there. It is anticoagulant. In a clinical situation, we may have to use this anticoagulant. And when you are preparing blood samples for biochemical analysis, often we are using blood coagulant, anticoagulant for preventing coagulation of the blood. Okay. I think I told you coagulant. That's wrong. This is an anticoagulant. Heparin is a natural anticoagulant. And how is it acting? This heparin inhibits blood coagulation by binding to the protein antithrombin. Heparin binding causes antithrombin to bind to and inhibit thrombin. You know that thrombin is essential for blood coagulation. Okay, what's the structure of thrombin? It has two monomeric units, and these monomeric units are highly sulfated. You can see here a sulfate is there, so this is highly sulfated. One of the monomeric unit is either glucuronic acid sulfate or hyaluronate acid sulfate. How do they differ? You see, this is the molecule, and here either the COOH is downward or the COOH is upward. If the COOH is downward, you see here we call it hyaluronate, and it is beta because this OH is upward, it is beta. So it is beta L hyaluronate. It is L configuration, not D. And if this COOH is upward, then we call it glucuronate. Of course, that also is beta because this OH group is upward. So one of the monomer is either glucuronic acid or glucuronic acid 2-sulfate. You see 2S means 2-sulfate. This sulfate is on carbon number 2. Or it is hyaluronate acid 2-sulfate. One of the component joined by a 1,4 linkage. Actually, this is a wrong statement. This is not a beta 1,4 linkage. This is an alpha 1,4 linkage. Okay. What is shown here is an alpha 1,4 linkage. Is joined with a, what is this? This is actually N-acetyl glucosamine. See, this was a glucose. It is containing two types of substitution. One is a substitution with an amino group at carbon number 2. So it is glucosamine. And there are three places where sulfate is a substituent. Position 2 actually is on the nitrogen atom, not on the carbon atom, ring carbon directly. Position 3, position 6. So glucose, N-sulfide, this is the N-sulfide, 3-sulfide, this is the 3-sulfide, 6-sulfide, this is the 6th sulfide. So glucose, N-sulfide, 3-sulfide, 6-sulfide, actually not sulfide, it is sulfate. Glucose, N-sulfate, 3-sulfate, 6-sulfate. This is the other monomer. In this figure it is beta glucose derivative. This is alpha glucose derivative because the OH group is downward. So alpha D glucose, N-sulfate, 3-sulfate, 6-sulfate in beta 1,4 linkage with the glucuronic acid or glucuronic acid 2-sulfate or hyaluronate acid 2-sulfate. This is the structure of heparin. Remember, it is used to prevent blood coagulation. And actually, it is not that much significant here. I wanted to show you the acidic sugars. Acidic sugars, N-acetyl neuraminic acid. N-acetyl neuraminic acid is a sialic acid. But the difference is that the amino group is present on carbon number 4, not on carbon number 2. And we call it neuraminic acid. Why neuraminic acid? On carbon number 5, there is no carbon number 6. Instead, there is an R group. That R group is CHOH, CHOH, CH2OH. So this is not glucose actually. This is neuraminic acid. N-acetyl neuraminic acid. And it is an acid because on carbon number 1, there is a carboxyl group here in the ring. And this is a derivative of neuraminic acid. And 9 carbon backbone is there. And it is a receptor on the host cells for influenza viruses, including SARS-CoV-2. It is a receptor on the host cell in our cells, in our body, on our target cells, on the membrane. We have this sialic acid. SARS-CoV-2 is identifying this sialic acid in order to ensure that it is the target cell, and it is found in N-glycans and O-glycans, etc. It has very important role in host parasitic interactions, microbe host cell interactions, cell interactions, etc.